Hello everyone. Today in this video, we are going to discuss few important questions from molecular basis of inheritance. Molecular basis of inheritance, the topic in which we study regarding the DNA, RNA, translation, transcription, replications, all these things we study under these topics. So in this today lecture, we are going to study only basics of only DNA. Okay, that is deoxyribonucleic acid. Okay, means we are going to discuss about the first nucleic acid is of two types. One is DNA and another is RNA. So we'll be discussing only regarding the DNA. Okay. These are the basics questions you must be knowing regarding this, but I want to start this video series with the basics. We'll cover basics questions also, tough questions also, every type of questions I want to cover. So in these lectures only the basic types of questions is covered, which you have to remember these points. Okay, now just I've told you nucleic acid. When you talk about the nucleic acid, the nucleic acid is of two types. Okay, that is DNA and RNA. Okay, DNA and RNA. And what is nucleic acid? It is the polymer of nucleotide, already mentioned in the question. It is the polymer of nucleotide. Okay, then nucleotide consists of, means what thing nucleotide is made of. Okay, options are nitrogenous base, pentose sugar plus phosphate group. Okay, phosphate group. Okay, now try to understand. See, when nitrogenous base combines with the sugar, that is known as nucleoside. Nucleoside. Okay, now when nucleosides combines with the phosphate group, that is known as nucleotide. Nucleotide. It means nucleotide consists of phosphate group also and nucleoside consists of nitrogenous base and pentose sugar. Means in nucleotide there is nitrogenous base also, sugar also, and phosphate group also. So our answer will be all. Okay, what is our answer? All is our answer. Next, how many types of nitrogenous base are present in DNA and RNA? Okay, DNA and RNA, they are the polymer of nucleotides. In nucleotides there is nitrogenous base, phosphate group, and pentose sugars. First we'll talk about nitrogenous base. Nitrogenous base on the basis of a structure is divided into two parts. One is purine and another is pyrimidine. Okay. In one group you'll find adenine and guanine, and in another group there is cytosine, thymine, and uracil. Uracil is not present in case of DNA. Okay, in case of DNA, adenine, guanine, thymine, and cytosine is present. Okay, and instead of thymine, uracil is present in case of RNA. Then in any one nucleic acid, DNA or RNA, four types of nucleic acid in this nitrogenous base is present. But the question is, when DNA and RNA both is given, then total how many types of nitrogenous base are there? 5. So our answer will be 5. Now, nitrogenous base, pentose sugar, and phosphate group. Then phosphate group, which is present in DNA, the characteristics of that phosphate group is asked. It is given they are negatively charged, acidic in nature, basic in nature, positively charged, 1 and 2 both. So phosphate group which is present in the DNA, that is negatively charged as well as acidic in nature. So our answer will be 1 and 2 both. Which bond is present between sugar and phosphate group in DNA? Okay, just I have told you nitrogenous base combines with the sugar and sugar combines with the phosphate with a bond. Regarding that bond they have asked: ionic bond, ester bond, glycosidic bond, coordinate bond, hydrogen bond. The right answer is that phosphate group combines with the sugar through the ester bond. Through which bond? Ester bond. Next, both polypeptide chain present in DNA is complementary, antiparallel, parallel, 1 and 2, 1 and 3. See, nucleotides, or you can say DNA, polypeptide chain, they consist of 2 polypeptide chains, this manner. Okay, here nitrogenous base are present on both the polypeptide chains. which are bond linked to each other. Okay, nitrogenous base. Suppose this side is purine and this side is pyrimidine on the polypeptide chain. They both this purine and pyrimidine are linked by hydrogen bonding. Okay, then both the chains are in opposite direction. Suppose this is 3 dash and this is 5 dash, then this will be 3 dash here and 5 dash here, but both are complementary to each other, and as you have seen, directionally opposite means anti-parallel. So our answer will be 1 and 2 both. Which nitrogenous base is not present in DNA? Adenine, guanine, thymine, cytosine, uracil. Just in the previous slide, I've told you uracil is not present in case of DNA. Cytosine binds with guanine in 2 polypeptide chain of DNA by 2 hydrogen bond, 3 hydrogen bond, 1 hydrogen bond, 4 hydrogen bond, none. Adenine, just I've told you in previous this previous slide regarding that in both the polypeptide chains, purine and pyrimidine are attached to each other by the hydrogen bonds. Adenine binds with the thymine by double bond and guanine with cytosine by triple hydrogen bond. Okay, that is 3 hydrogen bond. What is the length of helix length present in DNA? Okay, helix length, when you talk about this, I've told you helical structure is there. Then in this one complete how many steps are there of nitrogenous base? There are nearly 10 steps. Okay, 10 steps, and distance between one steps is 3.4 angstrom. Then of 10 steps, it will be into 10, that is 34 angstrom. Then that is known as helix length. Then total, what will be the helix length? In one step, there is 3.4 angstrom. Distance between two steps, C is 3.4 angstrom. And overall, in one turn, how much steps are there? 10 steps. Then 10 into 3.4, that is 34 angstrom. The helix length will be 34 angstrom. According to Chargaff's equivalency rule, purine equals to pyrimidine, purine greater than pyrimidine, purine lesser than pyrimidine, none, all. See, according to Chargaff's equivalency rule, both the polypeptide chain present in the DNA, in that case, purine will always be equal to the pyrimidine. These are the important points which you have to keep in your mind. Melting point of DNA depends upon adenine content, guanine and cytosine content, adenine thymine content, all or none. So if you talk about the melting point of the DNA, it always depends upon the content of the guanine and cytosine. So our answer will be guanine and cytosine. None is wrong. This is by mistake. The right answer is guanine and cytosine content. This is the right answer. This none is wrong. So guys, this I know there was not much more explanation at this point. These are the important points which you should remember, because this is also the syllabus of the Food Corporation of India Assistant Grade 3 Technical Examinations. So, and tomorrow in the video I'll bring little higher level questions. Slowly I'll increase the level of the questions and also with the proper explanations. Thank you.